Namaste students, हमेशा खुश रहो बिटिया. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth Women's University, Jaipur. So today we are going to discuss about the cell disruption, and this is under the subject upstream and downstream process. So what is cell disruption and why it is needed? So this is an integral part of the applied biology, specifically the veterinary and in the downstream process. So it is used to extract or to isolate the recover the product which are produced inside the cell. So it enhances the amount of product, means the recovery of product from the biological cells. There are three types of methods: mechanical, non-mechanical, and chemical or enzymatic methods. So uh, if cells are disrupted or the product is isolated from one method, then why another method is required? So why the various methods are existed? So the answer is because various types of cells are there, that's why various methods are there. So which type of cell is there? Is it bacterial cells, the animal cells, plant cells, fungal cells, etc. So each type of cell have its characteristics, its specificities, its speciality, and its susceptibilities. So according to that, we will choose the suitable methods. So what are the factors that affect the choosing of the various methods? Are the susceptibility of the cells, the product stability, the speed of the process, and the cost of the overall production. So let's start with the mechanical physical methods. Okay. So in this type of method, cells are being subjected to high stress, and the stress may be created by the pressure, the abrasion, with rapid agitation with beads or ultrasound. So because it is a physical or mechanical method, so heat generation is there, and that's why the cooling is required. Otherwise, your product may be lost. So first method under the mechanical physical method is the bead mill. So the diagram you can see, main principle requires a jacket grinding chamber with a rotating shaft running in its center. So this shaft is required to rotate the chamber. So agitators are fitted with the shaft and provide kinetic energy to the small beads that are present in the chamber. So as the shaft rotates, the chamber is also rotating, and the beads which are present inside, they have they gain the kinetic energy, and they collide to each other, and that make the choice of bead size and weight is greatly dependent on the type of the cells. So the collision is the generate the mechanical physical uh, phenomenon which is required for the disruption of the cell. So next is the ultrasound. So ultrasound vibrators that produce the high frequency sound with a wave density of about 20 kilohertz per second. So this is a unit of the ultrasound frequency vibrations. So here a transducer waves into transducer is there that converts the waves into the mechanical oscillations through a titanium probe. So basically, what is that? That there is a probe, a titanium probe, and uh, 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 the probe is converts the waves into the mechanical oscillations. Okay. So the ultrasonic vibrations are there. Okay. And the transducer uh, converts the ultrasonic vibration waves into the mechanical oscillations to the titanium probe. So titanium probe will oscillate, and the oscillation causes the disruption of the cells. So the bacterial cells and fungal cells are widely used for the ultrasound method for the cell disruption. The various type of cells require the various type of the frequencies and the various time periods, such as the bacterial cells require 30 to 60 seconds for the cell disruption, while the yeast between 2 to 10 minutes. Okay, so this method is usually used in combination with the chemical method. So if we want to lyse the bacterial cells, we will use the ultrasound with the lysis buffer. The chemicals are there which helps to cell disruption. Okay. So next method is the French press and high press homogenization. So and the cell suspension is drawn through a valve into a pump cylinder. Then it is forced under pressure of up to 1500 bar through a narrow annular gap and discharge valve where the pressure drops at atmospheric. So what is that? That you have the cell suspension, okay, and it is filled in a pump, wall pump cylinder, and in the cylinder it feels the pressure, high pressure, 1500 bar, and in this pressure the cell suspension is forced to discharge with a narrow valve or narrow pine valve. Okay, so uh, in the pump, in the cylinder, the cells feel the high pressure, and suddenly the cells are forced to discharge with a fine valve, and they feel the atmospheric pressure. So this sudden change in the pressure from the 1500 bar pressure to the atmospheric pressure, sudden drop in pressure upon the discharge causing the cells to explode. Okay, so the cells will explode, and it is mostly used for the yeast cell. It is a vital unit in the dairy production also industry, and the also used for the milk homogenization. So in the next lectures we will discuss the physical methods and the chemical and enzymatic methods. So thank you. Hope you are satisfied with this detailed session. If you have any query, please write in the comment box. I will resolve your query. This session is powered by Digital Version 2.